Next, I would like to, I have the honor of introducing Chip Aquadro. Let me make it so I can see my intro. Charles, by given name. Chip began his training at St. Lawrence University and the University of Vermont before heading to the University of Georgia, where he did his PhD. After completing his postdoctoral work in the Laboratory of Genetics at the National Institute of Environmental Health Sciences in the Research Triangle Park in North Carolina, he accepted a faculty position at Cornell. This was 1985, and he has been there since. So he has served on the faculty at Cornell for over 35 years. During his time at Cornell, Chip has explored a wide array of taxa in order to understand the interactions of evolutionary forces and genetic mechanisms in determining the role and signature of selection in biology. In the words of one nominator, Chip has made many of the most fundamental research contributions to the fields of population genomics and molecular evolution over the past 4 decades, and his work has been absolutely instrumental in shaping our current understanding of the underlying principles that determine the amount of diversity observed in natural populations. His work has ranged from experimental to statistical, but when we asked Chip about his greatest contribution to the field, in his opinion, it was the outstanding group of students and postdocs that he's had the opportunity to work with during his career so far. Will you please join me in welcoming Dr. Chip Aquadro to the stage?
Okay, I'm in. Okay, good. Okay, great. And you can hear me fine. Okay, good. Okay, now I've got to share my screen so that, oops, I need to share my window. PowerPoint share. Okay, can you see the slides? Great, good. Okay, well, I've already had this introduced, so I'll jump ahead to the next slide here. And I just made myself some notes because I get a little bit, flustered in this. And basically, I want to say thank you. I'm really deeply honored and flattered. I mean, the previous award winners of this award are just a who's who in molecular evolution and some of my greatest heroes. So I'm really honored to be included as part of that group. Frankly, it's also particularly gratifying because, as Sarah said, to me, this really honors the amazing undergrad, grad students, and postdocs I had the fortune to work with, and they really are, to me, my greatest contribution to science. And I've been fortunate to be in this field during a really exciting time. It started off with protein electrophoresis, and before first genome was sequenced of a virus when I was a graduate student, to now we can sequence whole genomes sort of just very quickly over and over again, as Molly so wonderfully pointed out. But it's been an extraordinary journey kind of for me, and I've loved every minute of it. It's been a remarkable group of colleagues and so forth. And frankly, the things to me is I feel really fortunate because I feel throughout my career, I have never had competitors. We have fellow grad students, fellow colleagues around the country at Cornell and other places. And we always counted on each other to call each other out on things and to bounce ideas and share ideas and things and feedback and constructive criticism. And to me, that's always been an incredibly rewarding part of my nearly 40 years in the career. Okay, Mike, time in biology started as an undergrad, and I was fortunate to have 2 amazing mentors. One shown here, Ken Kroll, together with my wife Wendy and I, is their place in Maine, and John Green. And both of them really stimulated my interest in biology and in research and gave me the flexibility to explore things both in small mammals, small mammal ecology, and also coyotes in the Adirondacks. I moved from St. Lawrence with a degree in biology to the University of Vermont to work with Bill Kilpatrick, who was a real mammologist and was really interested in using genetic variation to understand organisms. And I took from Ken Kroll's influence a project to actually look at variation in Peromyscus on islands in the islands of Maine, and showed that genetic variation was also correlated with levels of isolation and population size in those. So, and he really taught me to be a real mammologist. And so I went from there to obviously having learned to set traps all over the place and love the little mice Peromyscus that I worked with, to work with John Avis at the University of Georgia. And John was an amazing person. He was early in his career, actually, and he was most interested in speciation of fish, but was willing to take on somebody wanting to work on Peromyscus because they also shared an interest in birds with John. And I was there from 1977 to 1981 working my PhD. And it was a remarkably rich and diverse environment at that time. Headed up by Wyatt Anderson, John Avis, and Mike Clegg here, also some other people, Marjorie Asmussen, and so forth. And we had an amazing string of visitors that would come through, including Sewell Wright came for a period of time when I was a graduate student, a whole slew of others. And it was a remarkably strong group of population geneticists. And this is where I got my real sort of deep dive into population genetics. Through my first major course in population genetics in 1979, taught by Wyatt Anderson together with Marjorie Asmussen. This was also an incredibly productive time. John really taught me how to write scientific papers, and we wrote a lot of them. We also took advantage of the fact that John had a connection with a radio tower in Florida that collected birds that hit the tower, and they froze them, and we did an enormous series of papers studying the kind of phylogenetics of birds based on, in fact, protein variation. And this was, again, an incredibly productive 4 years, but it was also a time of enormous fun. We worked really hard, we played really hard as well. And here's the population genetics whitewater canoeing group, Wyatt Anderson, his wife Margaret, my wife Wendy, Doug Kavanagh, who's at Penn State now, Mike Clegg, and Jim Rawson, unfortunately, has passed away. But we spent many a wonderful weekend kayaking and canoeing in rivers in North Georgia. In 1981, I moved to the National Institute of Environmental Health Sciences to become a postdoc with Chuck Langley. Chuck, we had met the previous fall, and he had offered, said, why don't you apply? And I applied, and I got a position in his group. And this was in, again, Research Triangle Park, the one institute that's not in the major Washington, D.C., Baltimore area, Bethesda area. And this was an amazing group. And this is when I really, and Chuck gave me a lot of flexibility, I started really working on first making a transition because of my interest with John on mitochondrial DNA, diving into the first population genetic analysis of a sample of humans that Barry Greenberg had sequenced and several others in looking at sequence variation there. And again, this was truly done in the back of an envelope because there weren't that many polymorphisms. I shifted to Drosophila with my deepening appreciation of classical molecular genetics and population genetics theory, partly due to Chuck's enthusiasm and basically making the case for how important and how much information there was to be gained studying Drosophila instead of mammals. And I really found that to be very true. Even though John and I had a talk when I was a grad student, I swore I'd never work on Drosophila. He said I'd never write a book, and both of us broke those promises. He started writing a whole series of books, and I shortly thereafter moved to Drosophila. It was an exciting time. We were able to start to get glimpses of the nuclear genome in Drosophila using restriction fragment-like polymorphisms that really were spearheaded by Chuck Langley and a number of different genes during that time that I was there. And for me, the ADH gene was my real launch into that. It was an amazing time. Chuck really drew together a phenomenal group of people that included Dick Hudson and Marty Kreitman, Steve Schaefer. Wyatt came for a period of time. There were a whole series, Monsi Aguade, a whole series of people came through the lab and as well as worked there and together with Chuck and this incredibly powerful group. And including later Wolfgang Stefan shifting from physics like Philip Messer to into population genetics. And here's together with Chuck and Dick Hudson and Wolfgang Stefan at a molecular evolution meeting in 1969. As I said, there were some great opportunities with others too. Steve Schaefer came up and did his PhD thesis research in my lab on the alkyl dehydrogenase gene. Again, we were just running southern blot gels after southern blot gels, a small amount of space. We would work often double up our space of benches. Wyatt Anderson also came up and he and I cloned the amylase gene out of the Drosophila, a third chromosome inversion and pseudo-obscura, because he was really interested in the evolution of the inversion polymorphisms. And a series of papers emerged from that time there. Now, this was a time when there was a lot of debate regarding the role of genetic drift, random things, and versus natural selection. And particularly after the publications of Kura and King and Jukes, there was a lot of interest in fact, maybe that a lot of molecular evolution occurred by genetic drift. Interestingly, Dick Hudson and I both gave talks to the International Congress of Genetics in Toronto, and I was sitting in the audience when Dick gave his talk, and he talked about how he actually modeled Marty Kreitman's classic 1983 data on the alkyl dehydrogenase gene polymorphism in 11 alleles, and the pattern of variation Dick was able to model with a single site under balancing selection. And in fact, it was a remarkable fit exactly around the site where the allozyme polymorphism was. Well, I was fortunate to be sitting in the audience with Moto Kimura. I'd gotten to know him quite well, which was fun. And he leaned over to me and whispered, yes, only one selected site in 2,900 nucleotides. So he was still clinging to those few sites that were, the large number of sites that he viewed were neutral. So again, I just want to emphasize that a lot of people had a big impact on me, but I have to say Chuck had really the most dramatic impact on me in terms of shifting the direction of science, the way I did science, the kinds of approaches that I did, always thinking about what's the question and trying to frame my research in that way. And so I really feel like he had a dramatic effect on the field as well as a group of us in what I call the intellectual hitchhiking and Langley effect. So Chuck, thank you. So I moved in 1985 to Cornell, as Sarah had said, and in fact, here I was able to really start launching a wider group of studies, and in fact, trying to look at the interaction of natural selection and recombination. And I really started to focus on what made organisms different. And here's where I really kind of viewed myself as beginning to peek into the evolution's laboratory notebook much more deeply. And in fact, I was really intrigued by the footprints of natural selection. We could scan across genomes to look for unusual patterns, and we could also look at genes that evolved very, very rapidly as candidates for positive selection. And there were a lot of interesting motivations for studying variation and recombination to have an insight into what selection was acting. And in particular, several papers had pointed out this idea that, in fact, selective fixation of one variant can affect the frequency of linked polymorphic variants on either side of that fixed variant. And you can picture that in the following way. You start with a population here, all neutral variation at different frequencies. These are all sequences that have the same nucleotide as the top one. Differences down here. And if we take a look at this population sample, and if we have a new mutation occurs here that happens to lead to reproductive success, increased frequency of its being transmitted, that rapidly sweeps through the population to fixation. And it fixes that chromosome on which it occurred, so dragging with it all the other variation. This was a concept called a selective sweep that, as best I can tell, was first articulated and rating it themselves by Barry Agioka and Marty Kreitman in a 1991 paper, looking at levels of variation in very low regions of the genome. Chuck had already looked at a couple of these regions together with Wolfgang Stefan and Monsiaguade. We had looked at them in melanogaster in the tip of the X chromosome, and Andrew Barry and colleagues looked at it in the fourth chromosome. And all these regions showed very low variability, and we concluded that same publication years, Barry et al. Paper, that recent hitchhiking associated with a selective fixation in one or more of these genes in this region could explain the reduced pattern of variation. So Dave and I, and it was a paper by Dave Begun as a grad student, we decided to look more broadly at this. And Dave and I focused on moving away from the tip of the X chromosome where recombination was very suppressed, the slope was very low, out towards regions where rates of recombination of the slope of the line here started to increase. This is physical position, polytene band, and this is genetic map position. And as we the idea was that if we looked in regions of really high recombination, the selective sweep window would only be very small, whereas in low recombination, it'd be very big. So we might expect to see a gradient of variation. And we, in fact, were quite excited. We put together a paper based on some limited data and subjects, submitted it to Nature. In fact, and this was the plot from that original thing, wasn't a lot of data, but in fact, it showed a positive correlation. Well, of course, that was initially submission was rejected very quickly as too little data. So Dave and I took to summarize all the variation we could. A lot of people thought the variation among different loci was just evolutionary variance, chance variation. But we were curious about recombination. But how do you measure recombination? Well, it turns out Drosophila had a physical map, so to speak, by virtue of polyteins. And drawing on some studies by Lindsley and Sandler in 1977, they captured this as what we called coefficient of variation, the number of recombinants per cytological band per chromosome. And that allowed us to take 20 different genes from across the genome and plot them. And remarkably, we found a strong positive correlation. The higher the rate of recombination, the higher the level of nucleotide diversity. This is all assayed just by restriction site polymorphism at this point. Theory predicted that we should see the reduction variability by sweeps only within but not between species. And certainly, if we looked at that, we found no correlation between divergence of species and variability. So this supported very much the idea that there really was this positive correlation. We sent off a new manuscript to Nature a little while later, and it was fortunate to come out in early 1992. This turned out to be really quite a striking correlation that, frankly, has held up in lots and lots of organisms for a long time now. So it's actually been quite satisfying. And again, our logic was that as we got in regions of very low recombination, the sweep windows would be big, it would accumulate, and the effect would be quite impactful genome-wide. And I presented, in fact, this in a paper at the Taniguchi Symposium in 1991 in Mishima, Japan, which was a real delight. So we also were focused on asking single populations, not a group of different data. And so Eric Kindle in the lab took to look at the third chromosome of Drosophila melanogaster, a whole series of genes across here. And remarkably, the correlation was even stronger when a single population was looked at. And in fact, this was a time in my lab that was just a stunningly exciting time. A number of others joined the lab during this time. Michael Nachman, Dave Begun was right there, Martha Hamlin, Mike Ford, Tom Mitchell-Olds was on sabbatic for part of time, Katie Simonson. And we really dove into looking about diversity in other organisms. Michael Nachman tackled looking at human diversity across the X chromosome, sequence variability. And in fact, the results were kind of limited, but in fact certainly suggested there was a correlation in terms of polymorphism, but not divergence across in the human genome. At this time, we started to really shift away from restriction site polymorphism to full DNA sequencing. It was still done by autoreds, and Michael Nachman had the clever ideas. Well, let's not run them all individual things, A, G, C, T, next to each other. Let's actually group all the A's and all the G's and all the C's together. And so we started doing that, and that, in fact, worked really well. We still, though, read them off with a paper and pencil, read the sequences off, and we could score very quickly any variants that were there. And again, as I said, this pattern is held up across a huge array of organisms. I'm going to go a little bit quickly here because I'm starting to run low on time. But there was a question: could this really not be positive selection, but could it be the elimination of negative variants in the genome? And this was an idea that Brian Charlesworth really formulated quite well. Brian Charlesworth shown here together with Wolfgang Stephan and Chuck Langley. And he called this model background selection, or as he liked to call it, his BS model. And the concept was that a selective sweep is going to lead to a skew in the frequency distribution, but background selection just reduces the overall genealogy. And there were some hard things to distinguish selective sweeps from that. And a number of us studied different aspects. Maybe we actually showed lower variation in regions of on the X chromosome compared to autosomes. That was consistent more with sweeps than background selection. Chuck Langley looked at some other metrics and also argued towards it being really selective sweeps for many regions of the genome. But it was still clear that the biggest challenge was sorting out demography. So a target of a selective sweep predicts a very nice clean pattern of variability here in terms of reduction of variability right around the target of selection. The challenge, as Kevin Thornton and Jeff Jensen, with Jeff was a grad student in the lab, is that you can generate these exact same patterns simply by a severe population bottleneck. So this drove us, Jeff Jensen here, into a long period of studies of uh, trying to distinguish between selective sweeps and background selection and the influence of demography and teasing it apart. And it's still very much a major significant challenge in the field. I simply want to end very quickly with two quick stories about kind of actually taking some of these footprints and following them forward. And I'm going to focus on here first just one study by John Poole. John had done collections throughout Africa for Drosophila melanogaster and came back with a collection of remarkably variable Drosophila melanogaster in pigmentation. And remarkably, had really good records, and he was able to show that darker flies in individuals were present at higher elevations. He actually was able to map, and this is the actual data here, using patterns of sequence polymorphism, segments that were consistent with where the variation that accounted for this variation pigmentation was due to. And he was able to actually show that, in fact, there were 7 nucleotide variants, 6 of them in a regulatory region upstream of this gene called ebony, that in fact accounted for this variation. Mark Rebus and Sean Carroll joined in the effort, and they in fact did beautiful work to actually tease apart the pattern of cis regulatory evolution. We were able to say that there was a high ebony expression initially with standing variation that weakened the enhancer, but then there was dark mutations that were specifically selected, so new mutations to in fact lighten it even more. So in fact, it was a combination of selection on standing variation and new mutations that led to this pattern of adaptation that seems to be associated with altitude and various other factors. Finally, I just conclude with pointing out that we've also been focusing on reproductive genes. They're fascinating because they tend to evolve so rapidly. In many cases, it's thought to be due to conflicts between males and females, and a whole slew of people, including colleagues Mariana, Andy, Dan Barbash, have worked on this together with us over the years. And in fact, we've been able to actually show that that there's a remarkable amount of diversification. One of the things that struck us was we wanted to focus on something we could really try to tease apart functionally. And so we actually dove in on a gene that was involved in germline stem cell maintenance and differentiation. They don't have the premeiosis, they're genetically identical, no interaction with males and females, no competition. So a lot of the classic conflicts should be absent. And this gene called bag of marbles is the key switch that basically takes a germline stem cell and begins it down the path of differentiation. If you delete the gene, you get a bag of marbles. It just looks like this. It's just basically a tumorous germline stem cell. This gene, in fact, 442 amino acids, evolves enormously rapidly in certain lineages. And the question is, why? Why in the world would such an important gene evolve so rapidly? So we, in fact, have taken an increased functional approach to the analysis here and have focused on using CRISPR-Cas9 to generate null mutations across a series of different species. And to our absolute surprise, when we generate these nulls, in some species, it's still Jackie Bubnell here showing their first null here. In fact, we find that, yes, just like melanogaster, in fact, if we make a null, we actually get tumorous ovaries and testes. Remarkably, though, if we look at a closer related species, there's no effect whatsoever. In a more distant related species, we only see tumors in females, but not in males. So this suggests then that there's, in fact, an evolution and an evolution of function over time of this particular gene. And right now, our challenge is to try to match this bursts of sequence evolution with changes in function of this gene. And we also know this gene is involved in interaction with the Wolbachia, an intracellular reproductive parasite. So it's an interesting and fascinating time. And I simply let me totally end here because I think I'm just on overtime now. I think, let's see.
We need to wrap up.
Yep. Okay. So I just want to conclude by saying that again, it's been an amazing opportunity for me. I've had about 40 grad students and postdocs, taught population genetics for 29 years before turning it over to Philip Messer, and then also so far I've been teaching a course in personal genomics in medicine most recently. So it's been an amazing career, an amazing opportunity. And again, I want to thank the organizers of the meeting for all their efforts in this particular meeting. Congratulate Molly and Philip as well on their awards, and thank all of my colleagues for a wonderful career so far. Thanks. Bye.
Thank you so much, Chip. So we do not have time for questions, but I believe Chip will be around for the day, and we do have a break
Yep.
coming up. We have a 2-minute transition time now for switching to the Graduate Student Excellence Symposium. This is an SMBE tradition. As you may or may not know from the in-person meetings, this is a session devoted to graduate students presenting their work, and there's never any competing symposium at the same time. So I'd like to invite Harmeet Malik to come up to the stage. He'll be moderating the session for us. And if folks need to take a quick bio break, please do so, and we'll be starting promptly at 8:15.